Hello and welcome back. In this section's hands-on lab, we'll be looking into data from a publication a few years ago from Himes et al. On RNA-Seq analysis of airway smooth muscle cells upon treatment with a common steroid, a glucocorticoid steroid, used for the treatment of asthma. Now, this class of compounds, they've been known to have anti-inflammatory effects on one of the main muscles that line our airways, so-called airway smooth muscle cells, for some time, which is why, of course, they're found in many asthma inhalers and the like. Now, despite the fact that this activity has been known for some time, the actual detailed molecular mechanisms underlying the action of these compounds has remained somewhat unclear. It's not clear, for example, why some folks, when treated with these compounds, don't respond, why they seem to be non-responsive. There's low efficacy of these types of compounds for certain individuals. Now, if we knew more about the mechanisms of how they work and how they don't work in those individuals, we could potentially come up with a better type of therapy or improved types of compounds for the treatment of asthma, for example. So Himes et al. Set out on this road to try and understand these mechanisms, and they began to use RNA-Seq to profile gene expression changes in 4 ASM cell lines treated with dexamethasone, a common synthetic glucocorticoid used for the treatment of asthma. Now, they used top hat and cufflinks to quantify transcript abundances in exactly the same way we did actually in our last hands-on session, where we used Galaxy to coordinate this analysis. They focused in on just one gene that they found. Well, they actually found many genes that were differentially expressed or differentially regulated upon the treatment with these compounds, but they really focused the remainder of their manuscript on one gene called CRISP-LD2 that encodes a secreted protein that they had previously found was involved in lung development. In fact, they and others had found SNPs in CRISP-LD2 in previous GWAS studies that were clearly associated with inhaled steroid resistance. This bronchodilator response wasn't as effective for certain patients who had SNPs in this gene. And this data, together with this RNA-Seq data, strongly suggests that this is an important gene for mediating the effects of this class of drugs in asthma patients. So they went on very nicely to further confirm that there's differential expression of this gene. They used quantitative PCR, looked at increased expression levels with Western blotting, and did a really nice study to prove that this gene is indeed affected with these compounds. Now, we'll see in our hands-on session, this is just one gene out of many interesting genes that can emerge from these types of experiments. And they'll emerge if you know enough bioinformatics to dig in and get your hands dirty and pull out this kind of information. So that's what we're going to do. We're going to go back to where we left off the last day. That is the output of the mapping and the counting step here that these authors produced, just like we did the last day. And our starting point then is going to be this count matrix, where each cell indicates the number of reads originating from a particular gene. That will be the rows in this count matrix. For each condition or each sample, that will be the columns of this count matrix. Actually, it looks just like this, right? Recall from last day, it's a little table, or actually a big table in this case, because there are many genes, where we have the number of reads coming from each of those genes in the rows, and each sample here in the columns. For example, drug treated or control are the different columns here. Now, there is a vitally important second piece of information that we always need before we analyze data like this. This is called the metadata about the columns of the count data. So it's often called call data because it's telling us about the columns of the count data. That's the terminology used by many of the analysis programs that we're going to use. And this metadata is a small little table, just like this one here, that describes the columns. Right? It basically tells us about our experimental design setup, which columns in our count data are drug treated, for example, and which are control. Here is spelled out by this metadata table. Now, the vitally important thing here is that there must be a strict correspondence between the first column of this metadata, the call data here, and the sample names or the column names in that count data. So we need this correspondence in order to do our comparisons properly. Basically, we have to know which columns of data correspond to drug treated, for example, so we can compare the right columns here against the controls, right? If we don't, we're not going to be comparing the right things against the right other things. So to do these comparisons, we're going to need to install DE-Seq2. That's an R package available from Bioconductor, and we already discussed this installation in our previous video. The essential step being issuing these commands in your R console, not your R Markdown document, as we described. If you do it in your R Markdown document, you're going to potentially confuse yourself and lead to waiting a long time for these things to finish. So once we're set up and we have DE-Seq2 installed, we'll go through in detail how the underlying analysis works, so we have a good mental model for what's going on here. We'll actually first code it up ourselves from reading in the raw data and then calculating the log2 fold changes between drug treated and control, and we'll discuss each step in that process and why we use those types of units, for example. But once we have a good idea for how things work, we'll actually turn to DE-Seq2 to do this analysis more robustly. And again, we'll describe each of these steps shown here, from data input to setting up and running this DE-Seq analysis pipeline and getting the results. What we'll get out of this procedure here are results like this. Again, we'll have a row per gene, but this time we'll have data about the expression and the significance of this expression. As we've discussed, we'll have the log2 fold change here, for example, telling us whether the expression for a particular gene in the rows went up or down and by how much, and additional columns that report on the statistical significance of this change. Now, this is important, of course, because we nearly always want to focus our attention on the subset of genes that change a lot and whose change is significant between those conditions. And we'll talk about these columns and where they come from in our hands-on session. In particular, we'll focus in on this column, the so-called adjusted p-value here that are returned from methods like DE-Seq that are corrected from multiple testing. Now, this is important, as we've already seen. Of course, with genomics, we have always lots of data. And in these types of workflows like this one, we're typically going to be doing lots of hypothesis tests. For example, here, we have 20,000 odd genes here, and we're going to be doing 20,000 separate hypothesis tests. And if we used a good old standard p-value cutoff of 0.05, you'd expect 5% of that 20,000 genes to be deemed significant. That's 1,000 genes to be called significant just by chance, okay? So we're not going to live dangerously like that with that false positive rate. We're going to adjust or correct these values to control for the expected false discovery rate. Now, the simplest approach is so-called Bonferroni correction, that in essence involves dividing the regular p-value by the number of tests you're performing, basically rejecting any hypothesis with a p-value less than or equal to alpha over m, where alpha is typically 0.05. Now, DE-Seq2 uses a slightly more stringent Benjamin E. Hirschberg correction, and we'll talk about where that comes from and how we determine it in our lab session. At the end, what we'll arrive at is this happy intersection of fold change and significance estimates that keep both our inner statistician happy and our inner biologist happy too, right? Remember that, of course, fold change can be large, like twofold up or twofold down regulation, without being statistically significant based on those p-values, for example. And that small p-values for genes might not be relevant, right? They mightn't be relevant in terms of the biology. As biologists, regardless of whether that p-value is really, really small, indicating a very significant change, we're nearly always interested to know, which genes are most dramatically regulated, as these are often thought to be reflections of changes in biologically meaningful transcripts or pathways. So we'll show both fold change and significance together in plots like this one, sometimes called a Volcano plot. This is a common summary figure used to highlight genes that are both significantly regulated and display high fold changes. So each point here in this plot, we'll make these plots ourselves from the raw data up. Each point is an individual gene, and what we're plotting here is its fold change along the bottom axis. So further from the middle, that 0 point where there's no change, of course, means there are higher expression changes, either up in the positive direction or down in the negative direction. Now the side vertical axis here, this is minus the log of that adjusted p-value. Again, we use log values here to kind of spread out the values so we can examine them in more detail here, and we use minus because we're interested in small p-values. Remember that small values mean significant changes, so we're going to use minus of the log here. So now the most significant changes are going to be at the top of this figure, towards the high numbers here along this axis. So essentially what we're looking at are the subset of genes here that are colored up in blue that are exploding out from the middle of this volcano towards the top of the plot. So we'll make this kind of plot together, and then we'll go and we'll analyze the subset of the blue genes here, those that are both significant and change a lot, and put them into the context of their potentially more relevant biological pathways. And to do this step, to do this kind of pathway analysis, what we'll do is we'll take our subset of differentially expressed genes that we've just extracted, and we'll correlate them with our knowledge of pathways and biological processes. So in essence, this is a bit like, going to all those big textbooks that you had to purchase or get from the library for biochemistry and cell biology, molecular biology and immunology and the like, and all that accumulated literature, and then seeing which chapters in all those books or papers are lit up with our set of differentially expressed genes that we get from our experiment. Is it a chapter on cell division or intracellular signaling through MAP kinases or whatever that is most prevalent in our differentially expressed genes? So we take all those pathways with their constituent genes indicated by the blue circles here. Of course, we don't get them from textbooks, we get them from bioinformatics databases, and we get our differentially expressed genes. That's the gray circle here. This is our experimental results, our gene set, and we look to check for their overlap, or more formally, are the differentially expressed genes here enriched for a particular pathway or gene set from our set of available gene sets from these databases. Now the differentially expressed genes, they come from our experiment. The pathways, the genes, they come from annotations, not from textbooks, of course, but from bioinformatics databases like KEGG and Go, etc. And then there are variations in the way we can do the math and check for overlap and rankings or use networks, and how we calculate the enrichment and the significance of that enrichment. It can differ between different packages and tools, but in practice, the different algorithms show quite similar performance. We're going to focus in our hands-on session on a couple of methods available from packages within the bioconductor framework in R to do this gene set enrichment. Now, the differentially expressed genes, the ones that come from your experiment, they need to be as clean as possible. The pathways, they can come from different databases. We'll use a couple of common ones, including Go, the gene ontology, and KEGG, which is mostly metabolic and signaling pathways. But there are others, and which pathway you use or which database gene set you use is going to depend upon your research question in the future. Is it cancer gene sets that you're looking at, or is it metabolic sets or something else, right? That'll determine the types of blue gene sets that you would use here in these little circles. So with that, I'm going to finish this little video here, and in our hands-on session, we're going to perform this pathway analysis and make figures just like this one, where each box here is a gene. This is from an asthma pathway that seemed to be very prevalent in the experiment that we were analyzing. And again, the genes here, they're colored red if they're upregulated, or green if they're downregulated. And collectively then, when we put this all together, we can begin to get a rather detailed initial view of how airway inflammation can be perturbed with these types of steroids that we are analyzing in this week's lab. And we'll discuss some of the biology and some of the details here that were emerging from these experiments when we analyze this data together. So for now, I'm going to finish this video here and summarize our complete workflow to date as shown here. We go from raw reads at the top here through quality control in step 1, through mapping in step 2, and counting in step 3, down into differential expression analysis in step 4 here, and then gene set enrichment analysis to finish off with. So that's all I wanted to cover for this video. So please join me in our next hands-on session where we'll walk through these steps together and analyze this data together. And it just remains for me to say thank you for watching. I appreciate your attention, and I'll see you next time. Thank you.